Hey there. So, if you happen to be studying genetics at any point in your career, one of the main topics that always comes up is this idea of mutation, specifically in how mutations can impact protein functions in the long run. A mutation is something that can arise in a sequence as an addition or deletion mutation, which is pretty self-explanatory. Something got added or something was taken out. This can seriously impact the coding sequence of an mRNA strand and cause the protein to be non-functional in the long run. In this example right here, if you look, we can deduce that we have what is called a nonsense change, where as a result of adding an A in front of this phenylalanine, we have produced a stop codon here. We have either UAG, UGA, or UAA. In order to determine what the mutant sequence is, we need to look at the normal sequence. To start, our first two amino acids are the same, but the third one is now isoleucine instead of phenylalanine, so obviously the changes occurred here. What we need to do next is look at the mutant sequence and see what possibilities are available in the normal sequence to allow this mutant sequence to occur. If we look at the histidine up next, we can see its possible codon combinations and compare those to the possibilities in the normal sequence. So in this case, we have CAU or CAC. So if we look up here, we can see that if we were to add the A here, it would shift these over. So we would have the first letter of histidine being a C, the second letter being an A. But then the only possible one that we could get in the mutant sequence would be this C right here. So if we go down here, we've determined this codon combination right here. And what we would want to do next is now determine which possible combinations we would have in the stop codon as well. So the only one that we can really have is this UAA, because the only possible one that we could have next is this U from the threonine, and then these two A's from the lysine. So that would basically determine this side of it. And for this isoleucine, the codon combination does not really matter, so we can just choose whichever one we would like, and obviously the A has to come first. So from there, we basically determined our mutant sequence. So if we want to write it out, our mutant sequence is now AUG, UGG, AUU, because we added an A in front of phenylalanine, which is shifted over. Now we have CAC and UAA, which is our stop codon. This is your mutant sequence, and that is a general overview of how to determine mutations in an mRNA sequence.